our challenges. According to the last cancer statistics worldwide, including both sexes, breast cancer is the second malignancy in incidence worldwide and the fifth malignancy in mortality. But among women, breast cancer is by far the most common cancer among women worldwide. 1.67 million of new cases are diagnosed each year. More than 500,000 women die each year because this disease. Everyone has someone close to them who has had or has breast cancer. One woman is diagnosed every 20 seconds worldwide, and more than 3 women die every 5 minutes worldwide. Breast cancer is also the most common cancer in our country. 21,000 new cases are diagnosed each year in Argentina, and 1,600 women die for this disease each year. Unfortunately, our province, Mendoza, is among the 4th states in Argentina with the highest mortality. Breast cancer, most breast cancer and ovarian cancer are sporadic. That means that it occurs by chance with no known cause. Between 5 to 10% of breast cancer are hereditary. It involves some high-penetrant genes such as BRCA1 and BRCA2, and 20% of ovarian cancer has an inheritance pattern. There are a percent of familial breast and ovarian cancer which share some but not all features of hereditary breast cancer. These families have high risk comparing to the general population, but they don't show early age onset or inheritance patterns. BRCA1 and BRCA2 are the most common cancer in hereditary breast cancer, and the chances of developing this disease by age 70 is 1 in 10 whether the woman has normal BRCA gene, 4 in 10 where the woman has mutated BRCA2 genes, and 6 in 10 whether the woman has mutated BRCA1 gene. According in respect to ovarian cancer, the chances to develop by age 70 this disease when you have BRCA mutated is up 60%. What are our contribution in the past few years? First, to begin the register of hereditary breast cancer in our province and to create a very special DNA and tumor tissue bank. In 2013, National Cancer Institute found the Argentinian Familial Cancer Network called RACAF, and one of these aims was to register the cases of hereditary breast and ovarian cancer or other type of hereditary cancers in our country. Now, RACAF is formed by 55 health professionals, 60 public and private institutions in 11 provinces. In Mendoza, we have now 4 nodes of RACAF, the University of Hospital, Regional Center of Oncology, INVECU, and Central Hospital. And I want to take advantage to highlight the work as genetic counselor of Dr. Analia Vargas, who works in the University Hospital, and Dr. Alejandra Mampel, who is genetic counselor in the University in the University Hospital. Oh, it's very, very fast. And in the Regional Center of Oncology. In the period between 2016 and 2017, 289 patients received genetic counseling in our province, and last year more than 400 patients received this genetic counseling. What is our second contributions to motivate and accompany the drafting in Mendoza of the law 9055 to achieve full coverage of genetic studies and risk reduction measures? The sentence "from bed to the bedside" is very common used to mean what translational medicine significance. And one way to cross this bridge is to offer the genetic studies to all the women who has high risk to develop breast and ovarian cancer. In 2016, we met with Dr. Caron, this physician who works in the legislature of our province. And he listened our proposal and he wrote a draft of this law. And finally, on March 13 of the last year, our province is the first in our country to create an unit to full coverage of patients with genetic cancer and full coverage of all measures needed to reduce this risk of breast and ovarian cancer, such as bilateral mastectomy or bilateral salpingo-forectomy. What are our challenges in this research field? First, to improve genetic counseling for patients with clinical criteria for hereditary breast cancer and non-pathogenic mutations in BRCA1 or BRCA2 genes. And I want to take advantage to highlight the work of Alejandra and Dr. Silvina Nadine. Most of the hereditary breast cancer have mutated BRCA1 or BRCA2 gene, but there are other less common tumor suppressor genes which is involved in the etiologenesis of breast and ovarian cancer, such as P53, P10, PALB2, and others. But there are a lot families when we study a multi-gene panel to know what gene is mutated, but there are all normal tumor suppressor genes normal. And these families belong to the BRCA-X syndrome because no one knows what is the real or molecular cause of this disease. For example, these patients, it's a very young woman, very, very young with breast cancer, who has her father with breast cancer, the sister of her father, and the mother of her father with the same disease, but all tumor suppressor genes studies are normal. And this is a situation very, very difficult to the physician because no one has molecular evidence to recommend measures to diminish the risk to develop breast cancer. What is our hypothesis according to this? This kind of patients with high clinical criteria of hereditary disease without pathogenic mutations in these or in other genes have a defective repair of DNA damage caused by ionizing radiation. When, for example, ionizing radiation causes a damage in the DNA, it produces double-strand break of DNA, and the homologous recombination is the DNA repair mechanism to resolve this double-strand break. There are several molecules involved as sensor mediators and effectors in this kind of mechanisms of DNA reparation, and BRCA1 and BRCA2 and the histone 828X are very, very important to repair these double-strand breaks. What is the methodology that we are applying now? First, we take a blood sample from the patient with pathogenic mutations, and patient with high criteria but without pathogenic mutations. And in INVECU, in the Laboratory of Tumor Biology, we culture the lymphocytes. Then we irradiate these lymphocytes in the Nuclear School of Medicine. I want to thank Dr. Sergio Viña for his help. And then the lymphocytes in our lab are divided into parts. A half is harvested immediately after irradiation, and this is the time 0, and the other half is recovered during 24 hours. This is our T24. And we can study by different techniques, alkaline comet assay, micronuclei, and foci of gamma 828X. What is the percentage of DNA damage, and at 42 hours, what is the percentage of DNA repair? We don't know what is the gene which is mutated, or probably it can be an epigenetic modification, but the most important, or our goal, is to provide the physician with who person don't repair the DNA damage, and she has a very, very high risk to develop the disease. The other challenge is to improve the targeted therapy against PARP in breast cancer with BRCA1 and BRCA2 pathogenic mutations. In this project works very, very hard Mayra Sotile, our postdoc fellow, and the general goal of this project is to establish if the simultaneous administration of olaparib and doxorubicin increases the individual sensitivity of these drugs in breast tumor cell lines with and without pathogenic mutations in BRCA1 or BRCA2 genes. Olaparib is the first human PARP inhibitor. It's the first personalized therapy against ovarian cancer. It's approved by FDA in 2014 in the treatment of ovarian cancer, and last year FDA approved the administration of olaparib in breast cancer patients with mutations in BRCA1 or BRCA2 gene. PRAP is a very, very important protein involved in the base excision repair, which is a DNA repair mechanism of single-strand breaks. And we said that BRCA1 or 2 are very important proteins in the DNA repair mechanism called homologous recombination. If we block, or if we applied olaparib to inhibit PRAP, the single-strand break during that time are converting in double-strand break. And if these cells don't have the homologous recombination because BRCA protein is not functional, you have a lethal synthetic lethality of the cells. For this deficiency in base excision repair, inhibiting PRIP together with non-functional BRCA proteins leads to the synthetic lethality. And we have to know what is the effects of simultaneous administration of olaparib and doxorubicin. Doxorubicin is an antineoplastic drug very, very used as the first line in the breast cancer treatment. But what is the problem of doxorubicin? It's cardiotoxicity. Sometimes a woman with breast cancer died for the cardiotoxicity and not for the breast cancer. For this, it's too important to treat to diminish the doses of doxorubicin. These are the first preliminary results of Mayra Sotile. She transfected these mammary tumor human cells with short heparin directly against PRAP1, and in these cells, which don't express these very, very important proteins, when these cells are incubated with increasing doses of doxorubicin, these cells are more sensitive. Probably in the future, with other experiments, we will offer a very good combination in breast cancer, diminishing the doses of doxorubicin. Finally, I want to acknowledge National University of Cuyo, National Cancer Research, and National Cancer Institute of Argentina for the support. I want to thank all my lab mates, Laura, Mayra, Silvina, Marina, and Analia. And last but not least, I want to thank all women who every day fight against breast and ovarian cancer and accept to participate in our research. Thank you for your attention.